So, as you remember from the first lecture that we did on reproduction, we said that the sex organs consist of 3 sets of structures, and these 3 sets of structures include the gonads, the internal genitalia, and the external genitalia. So let's identify these 3 sets of structures in the male reproductive system. So the testes are the male gonads, or the gamete producing structures in the male reproductive system. Then the internal genitalia, remember that internal genitalia refers to all of the accessory glands and the ducts which connect these gonads with the outside environment. So the male internal genitalia includes the prostate gland, which is over here surrounding the urethra, the seminal vesicles, which are over here, the vas deferens, bulbourethral glands, which are over here, as well as the epididymis, which is over here. So the epididymis actually becomes the vas deferens. So the epididymis actually lies on the surface of the testicular capsule. It's not labeled over here, but here you see the epididymis lying on the surface of the testes. So the seminal vesicles and the bulbourethral glands, they empty their secretions into the urethra through these ducts. So that's how they empty their secretions, through ducts. But the prostate gland on the other end, actually each of the individual glands of the prostate opens directly into the urethra. So secretions can be secreted into the urethra. So the urethra then serves as the common passageway for both urine, which comes from the bladder, and for the semen. So it collects all of the secretions from the different glands, and then it serves as the common passageway. So moving on to the male external genitalia. So the male external genitalia consists of the penis and the scrotum. So the scrotum is this external sac-like structure which you can see over here. So in other words, this structure lies outside of the abdominal cavity, and it's the scrotum which holds the testes. So in other words, the male gonads. Now the reason why the scrotum is located outside of the abdominal cavity is because normal sperm development, which obviously is happening within the testes in the male gonads, requires a temperature which is below the core temperature. So in other words, what we're saying is that spermatogenesis is actually very temperature sensitive, and for spermatogenesis to complete, it actually needs a temperature which is lower than our core body temperature, and that's why the scrotum with the testes is located in the external environment outside of the abdominal cavity. When we take a cross section through the penis, we see that the urethra is actually surrounded by this spongy column of tissue known as the corpora spongiosum. And then there are 2 more columns. These 2 columns are known as the corpora cavernosa, and the corpora cavernosa actually constitutes the erectile tissue of the penis. Taking a closer look at the testes, we see that they contain these coiled tubules, and these tubules are known as the seminiferous tubules. Now, these seminiferous tubules are clustered into about 250 to 300 compartments, and basically make up the mass of the testicles. And in between these tubules, you can't really see in this picture, but in between these tubules, the interstitial tissue is made up of the blood vessels and the Leydig cells. So very important to remember that the Leydig cells are not actually located within the seminiferous tubules, but they lie in between in the interstitial tissue. Now, as I said already, these seminiferous tubules make up the mass, or the most of the testicle, which is about 80% of the testicles, and each individual tubule actually varies in length, and this length ranges between anything from about 30 centimeters to 1 meter. And if these tubes are actually stretched out and put end to end, it would probably stretch over a length of about 250 meters. Now these tubules leave the testicles, and then join the epididymis. So this whole part is the epididymis. So you can see the epididymis is this tightly coiled cord which then lies on the surface of the testicular capsule. And the epididymis then goes on to become the vas deferens. The seminiferous tubules are the actual site of sperm production. So it's within these tubules that the sperm development and the entire spermatogenesis process is taking place. So looking at a cross section of the seminiferous tubule, we see that it actually contains two cell types. So firstly, the Sertoli cells. So the Sertoli cells, so this entire purple large cell is one Sertoli cell. So we see that the Sertoli cells extend from this outer edge of the tubule all the way to the lumen. So this entire cell, or this entire part, is one cell. This entire purple structure is one cell. And the surrounding, or the neighboring Sertoli cells are linked by tight junctions. So all of these different Sertoli cells are linked by tight junctions. The other cell type that is contained within the seminiferous tubules are the spermatogonia. So spermatogonia is just a plural of spermatogonium. Remember, these are the germ cells. But what we see is we see the spermatogonium, or the germ cells, in their various stages of development. So throughout the spermatogenesis or the gametogenesis process, we see these spermatogonia in their various stages of becoming sperm. So primary spermatocytes, and then spermatids, and then finally completely developed spermatozoa. Taking a closer look at sperm development within the seminiferous tubules, we see that the developing spermatocytes stack in columns. We can see that these columns stretch all the way from the outer edge of the tubule all the way to the lumen. And as we mentioned in the previous section, or the previous slide, between these columns are these Sertoli cells. Remember, stretching all the way from the edge all the way to the lumen. And this is very important because the Sertoli cells are vital in regulating the entire process of sperm development. So the Sertoli cells regulate sperm development, and they provide the necessary proteins and nourishment and support for the sperm production process. So starting from the basal ends of the Sertoli cells, so starting over here with the spermatogonia, at the basal ends of the Sertoli cells is where the spermatogonia undergo their mitotic division. So this is where they're producing additional germ cells. So just duplication of a daughter cell, which is exactly identical to the parent cell, just normal mitotic divisions. From there on, some of these spermatogonia then enter meiosis. So the spermatogonia that enter meiosis then become primary spermatocytes. Remember, primary spermatocytes contain 46 duplicated chromosomes, so they contain double the amount of DNA compared to the spermatogonia. Now, as these spermatocytes then differentiate and start developing into sperm or become more mature, they move inward towards the tubule lumen. So as they go through this whole process of meiosis, they move inward. So, but still continuously being surrounded by the Sertoli cells, these nourishing Sertoli cells. Now, by the time the spermatocytes actually reach the luminal end of the Sertoli cells, so the side closer to the lumen, they have then undergone both meiotic divisions and they have become spermatids. Remember, one primary spermatocyte forms 4 spermatids. So here you see the 4 spermatids. These spermatids then stay embedded in this luminal side of the Sertoli cells while they complete their transformation process into the mature spermatozoa. So the flagellated spermatozoa. This entire sperm production or sperm development process, so from germ cell or spermatogonium all the way to a mature sperm or spermatozoa, takes about 64 days. Now, throughout the seminiferous tubules or different regions of the seminiferous tubules, there are different staggering developmental stages. So in other words, at any given moment, a different region of the tubule contains the spermatocytes in their different stages of development. So in other words, throughout the seminiferous tubule, you'll have primary spermatocytes, you'll have secondary spermatocytes, you'll have spermatids, etc., etc. Now this allows for sperm production to remain at more or less a constant rate, which is about 200 million sperm per day. So 200 million sperm per day sounds a lot, but actually that's the amount of sperm which is released in a single ejaculation. Now during this transformation process from a spermatid into sperm, the spermatid actually loses most of its cytoplasm and it then develops a flagellated tail, obviously because it's a motile gamete. The chromatin of the nucleus also condenses into a dense structure, and this dense structure then fills the most of the head of the spermatozoa. Then also a lysozyme-like vesicle called an acrosome flattens out to form a cap over the tip of the nucleus. Now this acrosome plays a very important role in fertilization, as we'll see in the last lecture of reproduction. And then mitochondria is also concentrated in the midpiece of the sperm body. Obviously, mitochondria is needed because it produces energy for the movement of sperm. And the final result is this small motile gamete, which actually bears very little resemblance to its parent spermatid. So as I've mentioned already, the Sertoli cells play a vital role in the regulation of sperm production and development, because they are the ones that provide the necessary nourishment and the necessary proteins, etc., for these columns of developing spermatogonia. They manufacture and secrete proteins, as I've already said. These proteins include inhibin, activin, certain growth factors, various enzymes, and they also secrete androgen binding protein, or ABP. So ABP is secreted into the lumen of the seminiferous tubules, where it then binds to testosterone. And if we think back to the chemical characteristics of testosterone, we know testosterone is a steroid hormone. Being a steroid hormone, it means that testosterone is lipophilic, so it usually easily diffuses out of the cells. But when APB, which is a protein, binds to testosterone, it makes testosterone less lipophilic, which means that testosterone is inhibited or prohibited from diffusing out of the tubule lumen as easily. So it keeps testosterone within the lumen of the tubule. Now we move on to the Leydig cells. So we haven't really spoken much about the Leydig cells, except where I mentioned earlier that the Leydig cells are actually located in the interstitial tissue. So the Leydig cells not located within the seminiferous tubules, they are located within the interstitial tissue which surrounds the tubules. The function of the Leydig cells is to secrete testosterone. And these Leydig cells actually go through these phases of being more active and less active, or quiescent. So they first become active in the fetus when testosterone is needed to direct the development or the differentiation of the male characteristics. So remember that if we think back to the differentiation of the internal genitalia, we said that testosterone is needed for the development of the Wolffian ducts to become the male accessory glands and structures, and testosterone is also required for the migration of the testicles into the scrotum or into the scrotal sac. Then after birth, these Leydig cells become quiescent or inactive for a phase until puberty. So only at puberty do they resume their activity to then produce testosterone.